to do over the next few minutes is to answer a commonly asked question, namely, how could it be possible that you went from discovering a new virus in January of 2020 to approximately 11 months later to actually have a vaccine that goes into the arms of individuals in December of that same year, when we know generally vaccines take multiple years to develop? And this has often led to some degree of hesitancy on the part of people wanting to get vaccinated. So I'd like to answer that question. Yesterday, Science magazine published an editorial that I wrote, which tried to give an explanation of the story behind the vaccines. And the bottom line was that the speed and efficiency with which these highly efficacious vaccines were developed and their potential for saving millions of lives are due to an extraordinary multidisciplinary effort involving basic preclinical and clinical science that has been underway out of the spotlight for decades before the unfolding of the COVID-19 pandemic. Next slide. One can look at vaccine development in two major buckets: platforms and immunogen design. Next slide. With regard to a platform, what do we mean by that? Next slide. A platform is the type of vaccine that you use. There's genetic immunization, such as DNA and RNA, and hence the mRNA vaccines, viral vectors, such as the adeno with J&J, recombinant protein, such as Novavax, and a number of others shown on the right-hand part of the slide. Next slide. With regard to the mRNA vaccine, this work started a decade and a half ago when Caitlin Carrico and Drew Weissman were working on RNA and was able to modify the molecule to remove a key inflammatory component, which would have made use of this as a vaccine impossible. This rather transforming accomplishment of theirs led to the use of mRNA as a platform. Next slide. With regard to adenovirus, the Ad26 that's used by J&J, the NIH has been funding for decades a highly skilled and accomplished investigator, Dan Baruch, from Harvard Medical School, who have been working on this for a considerable period of time and demonstrated how immunogenic this particular vector would be. Next slide. And then you get to immunogen design, really the most fascinating component of this story. And it goes to a different discipline. Next slide. And that is the crystallography and cryo-EM capability of investigators throughout the country who were working to get the right conformation of the HIV envelope trimer. Nothing at all to do with coronavirus. They were using these technologies to get the most immunogenic conformation of the molecule to develop an HIV vaccine. Next slide. This work was done with great intensity also at the NIH Vaccine Research Center. And I've circled two investigators on this slide. The one on the lower left is Peter Kwong, who is predominantly an HIV investigator. And the one in the upper right is Barney Graham, who is much more interested in respiratory syncytial virus. Next slide. What Peter Kwong did is that he used this structure-based vaccine design to get the right conformation of the HIV envelope in what's called a pre-fusion form, which would be the most immunogenic and could bind to broadly neutralizing antibodies. Next slide. When Barney Graham took his interest in respiratory syncytial virus and collaborated in the same lab with Peter Kwong and Jason McClellan, they had a major breakthrough in respiratory syncytial virus vaccinology by developing mutations that stabilized the immunogen to make it be used successfully for an RSV vaccine. But the story goes on. Next slide. When MERS came about, they did the same thing in an attempt to develop a MERS coronavirus vaccine, namely taking the spike protein and stabilizing it by mutations into a pre-fusion form. And then the culmination of the story, next slide, when we had SARS-CoV-2 with Barney Graham and Kizzy Corbett and others in the laboratory were able to make a stable component of the pre-fusion spike protein. Why is that important? Because that's a highly immunogenic protein, which has been used in 5 of the 6 vaccines that we're currently involved with. Next slide. And so on this last slide, what you see on the left are the 3 platforms, the nucleic acid mRNA, the adenovirus, and the recombinant protein. And with that, we already have 3 highly successful vaccines that have a high degree of efficacy and a good safety profile. So the bottom line is, this did not happen in 11 months. It was due to an extraordinary multidisciplinary effort involving basic clinical and preclinical science that had been underway out of the spotlight for decades before the unfolding of the COVID-19 pandemic. And with that, I'll pass it over to Dr. Murthy.